Then you mute the YouTube and
great. Okay, is this streaming?
Yep. Mute the YouTube. Yep. Okay, mute the YouTube. Yeah, and then play.
Okay, perfect.
Then it'll catch up to us.
Okay, very good.
Yes, we're good.
Okay, you can see it?
Yeah.
All right. Okay, we'll just give people 2 more minutes here to sign in. Yes. All right, so it is 2 o'clock, so we're going to get started here. Welcome to the Philip J. Currie Dinosaurs Museum's virtual speaker series for April 2022. If you have questions during the course of the presentation, feel free to add them to the chat on the side of the YouTube there, and we will get our speaker to address them afterwards. We are very happy today to welcome our April speaker. Ashley Reynolds will be speaking to us about Ice Age mammals. So Ashley Reynolds earned an honors BSc from the University of Toronto in 2015 with majors in ecology and evolutionary biology and biological anthropology. Before joining David Evans' lab at the University of Toronto, she completed an internship in science communication with Discovery Canada, where she worked with Daily Planet, Canada's only daily science news, and her discussion topic. Her PhD thesis investigates bone histology, growth, and life history in felids, with a focus on extinct saber-toothed cat Smilodon. And so with that, I would like to welcome Ashley, and she's going to share her presentation and we will get started here.
Yeah, thank you. Thank you all for coming and giving us a watch. Let me see, my screen is deciding that it doesn't want to share. Give me 1 second. It wouldn't be a Zoom meeting if there's not a technical difficulty. All right, there we go. So just before I get started, I do want to acknowledge the land that I've been lucky to work on since even before I started my PhD. So for thousands of years, the University of Toronto and Toronto more broadly has been the traditional land of the Huron-Wendat, the Seneca, and most recently the Mississaugas of the Credit River. Today, Toronto is still the home of many Indigenous peoples from across Turtle Island, and I am very grateful to have had the opportunity to work and play and live on that land. So I'm going to be talking to you today about the finds that myself and colleagues have started working on relatively recently relating to Ice Age fossils that have been found around the city of Medicine Hat, so over in Alberta. And before I get into that, I do want to give you a little bit of a backstory about how I came to work on Ice Age mammals of Alberta. That was not sort of originally what I intended to do, and I'm still happy that I've managed to make it there. But my introduction to Ice Age mammals, sort of generally, other than, watching the movie Ice Age, was this beautiful cat that you see here. This is called Smilodon. It's a saber-toothed cat that lived during the late Pleistocene, which most of us will colloquially have known as the Ice Age. And Smilodons, so these saber-toothed cats, are members of the cat family, but they belong to a completely separate subfamily, so a completely different lineage of cats than all of the cats that we are used to today. And though that subfamily, that lineage of cats, is what we call the saber-toothed cats. Cats themselves are part of the mammalian order Carnivora, which includes basically all of the big carnivorous mammals that you can probably think of. So it includes cats, hyenas, dogs, bears, seals, weasels, and many more interesting creatures. Within that clade, or that group of saber-toothed cats, Smilodon is just one of many types. And within the genus Smilodon, we actually have 3 different species. But if you hear me saying Smilodon, I am referring specifically to Smilodon fatalis, which is the mostly North American version of this species that lived closest to when we are now. So it's one of the more recent species of saber-toothed cat. And as Emily said, my PhD thesis is about learning how Smilodon grew, and then comparing this with the growth patterns seen in living cats. So to this end, I am hoping to answer some age-old questions about Smilodon, some questions that have been sort of in the works for decades. And those include things like, did it live in social groups like a lion, or was it solitary like most living cats? And also, how sexually dimorphic was it? Were the males quite a bit bigger than the females, or were they more close in size? And some of the things that I've been able to find so far suggest that Smilodon had an interesting family life compared to what we expect by looking at cats today. So in 2021, myself and my colleagues published a paper in iScience that described 3 individuals of Smilodon from Ecuador. Based on size and taphonomy, we were able to suggest that they were likely a mother traveling with her cubs. And these cubs were relatively old, so they were probably better classified as teenagers than as cubs. And what this told us, or what this suggested to us, was that they stayed with their mother for quite a long period of time, which is something that we see in social living cats like the lion today, but not necessarily in solitary cats, which tend to become independent from their mothers a little bit younger. That said, while we found this sort of this indication that they grew, or they matured slowly, we also had evidence that they grew quite quickly, which is sort of the opposite. It is more similar to a tiger than a lion. So part of the remaining part of my PhD thesis is looking to see if there are other lines of evidence that might support those conclusions. And the major way I have been doing this is using a technique called bone histology. And this is a technique that has been more and more widely adopted by paleontologists over the last couple decades. And it basically relies on the presence of growth marks in long bones. And you can see those on the slide, or the photograph on the right of this slide. All of those green arrows are pointing to these growth marks, and they're sort of analogous to tree rings in the sense that they are deposited approximately once every year. And because of that, they can be used to estimate the age of an individual at its time of death. And in addition to this, because of the fact that they retain the shape and size of a limb circumference at the time that the growth mark was formed, these can actually be used to estimate body mass through an animal's lifetime. And because of these two things, because of the fact that we can get age and body mass together, if we have a good enough record of growth marks, we can actually create something called a body mass growth curve. And basically what that does is it tells us how big an animal was at different points in its life and allows us to compare that with animals today, for which we can much more easily get these body mass growth curves. So the way that we kind of get all of that information, the way that we get these body mass growth curves is by making a thin section of bone. And essentially what that entails is taking a saw to a bone and grinding it really, really, really thin until it is about the thickness of two human hairs kind of stacked on top of one another. So this is usually done with a weight-bearing bone, like a femur. You can see that's a thigh bone. You can see that's what I'm cutting here on this particular slide. And then once the thin section is made, I will look at it under a microscope. The light will pass through it and it will illuminate these growth marks. I can then trace the growth marks and determine how many there are, as well as how big they were at different time periods. So because of this, we do now know more about how Smilodon grew. So we have a better understanding of the fact that it was a very large cat. It was about 40 kilograms bigger than a lion, and it grew up more slowly than a lion. So it grew up within about 4 to 5 years. And what's interesting is that while it sort of seems more extreme than a lion or a tiger on all metrics, this is actually not unusual given the fact that it was a large animal. So if we compare it to other animals, we find that they tend to grow faster and they tend to grow longer the bigger they are. So what we're seeing in Smilodon is not necessarily a unique pattern. It's actually just an indicator, or it's just a byproduct of the fact that it is such a large animal. Now, most of the specimens that I use to do all of that work with the bone histology that I just showed you, they come from the Rancho La Brea tar pits in Los Angeles, California. And the tar pits are a really great resource because they preserve millions of fossils, the majority of which come from large carnivores like the direwolf or like Smilodon. And these species are usually quite rare in the fossil record. They're usually found at low densities. That is completely not the case at Rancho La Brea. And to give you a sense of just how prolific the Rancho La Brea deposits are, I've squinted a lot at this photo, and almost every single bone that you can see here is from a saber-toothed cat. So there are millions of specimens and thousands of individuals that come from this site, which is a really great resource. But not all sites are like that. And in my first couple of years working on my PhD, I discovered that Smilodon does actually have an interesting connection to Canada, which is my home country. It is the place where I have grown up and always lived. And I discovered this connection thanks to the fossil that you can see in my hand here in this photograph. And that fossil came from Medicine Hat. So as you all no doubt know, Medicine Hat is in the southeastern part of Alberta. You can see it indicated by the star on this map, and it's quite close to both the Saskatchewan and the Montana borders. This area where it was found is Treaty 7 land, so it's the traditional territory of the Blackfoot and the Métis people. And in the late 1960s and early 1970s, Rufus Churcher from the University of Toronto and Archie Stalker from the Geological Survey of Canada conducted a field program that was aiming to look at the geology and the vertebrate paleontology of the Canadian prairies. One of the areas that they spent a lot of time in was along the South Saskatchewan River in and around the city of Medicine Hat, Alberta. And in this area, this relatively small geographic region, they collected over 1,000 fossil specimens from about a dozen localities. And then these specimens were finally donated to the Royal Ontario Museum in 2014. So the fossils in the Medicine Hat area come from a series of rock layers that encompass the end of the Pleistocene, which, as I mentioned earlier, can also be called the Ice Age. Although, if you want a fun party fact for people, we are technically actually still in that Ice Age. We just happen to be in a relatively warm period within that Ice Age. So these deposits cover the end of the Pleistocene, so the last cold period during the Ice Age, and then into the Holocene, which is that warmer period that we're in right now. About 99% of the material that we have accessioned at the Royal Ontario Museum is from mammals. And you can see here a list of all of the types of mammals that are included in the collection. And what we notice is that most of these are actually species that we would expect to find in Alberta today. So many of these species are still living, but there are a few notable Ice Age taxa that are highlighted here on the slide in green. This includes many notable Ice Age herbivores like extinct camels, horses, mammoths, as well as different types of ground sloth. But what, of course, really drew my attention, being a cat lady, is this entry here, which is the cats, especially the Smilodon, because that's what my PhD thesis was about. And this was really remarkable to me because, as far as I knew, there were no Smilodon fossils that had been confirmed from anywhere in Canada. So I started digging a little bit into the scientific literature and found that this find, this Smilodon from Alberta, was only really documented in what are called fauna lists. And these are essentially just a laundry list of the species present at a fossil locality. These lists can be very valuable because they give you insight into what an ecosystem looked like at a particular point in time. But the standard in paleontology is that specimens should be described, and particularly they should be associated with catalog numbers of the specimens that those identifications are based off of. Having those data means that in the future, researchers can always revisit the exact specimens that the presence of a species is based on, and they can update the research to agree or disagree based on new information that we might get from elsewhere and other specimens. And this sort of importance of having the catalog number as well as having a specimen described is especially true for cases like this, where there's only one record of a particular species or a particular group of animals in a broader geographic region. So here on the slide in front of you, you can see a map of North America, and it shows all of the places that this species of Smilodon has been documented. And the red dot, of course, is Medicine Hat, Alberta, which is about 1,000 kilometers north of the next most northern locality that this species has been described from, which was in Idaho. So because of the importance of these possible Smilodon specimens from Medicine Hat, we decided to re-examine all of the cat fossils from Medicine Hat, confirm their identifications, and publish full descriptions of them. So the first specimen that we looked at was a tooth that was originally identified as a Canada lynx. When we measured the tooth, we found that it was about the same size as a recent Canada lynx, so that sort of tracks. But we also found that there is evidence that bobcats were larger in the Pleistocene than they are today. So because of the fact that size alone may not be a very good way to differentiate lynx and bobcats in the past, and the fact that bobcat and lynx teeth look very similar, we felt that it was safest to say that this was a cat in the genus Lynx. So it was within the lynx group of animals, but we couldn't determine whether it was a Canada lynx or a bobcat. So that was the smallest of the species that we found. We then sort of looked into the big cats that we had from Medicine Hat. So our first specimen here, or our first couple specimens, I should say, are a fourth metatarsal. So that is a toe bone that was originally identified as coming from the American lion, which is Panthera atrox. And when we compared these with other specimens in our collection from Panthera atrox, we found that they were remarkably similar in both size and shape to other specimens of the American lion. One of the ways that you can tell this, and the reason why size is so important here, is that American lions were gigantic cats. So you can see in this photo on the left here, you can see me standing next to the silhouette of one, as well as the silhouettes of some other cats. So I'm about 5 feet tall. I'm not that tall, as in not very tall at all. But you can see that the American lion dwarfed me, and it was probably, it was at least double what I weigh, maybe definitely more than, it was actually probably closer to 3 times what I weigh. So it was a very, very, very big cat. Now, American lions have been found all over North America, including in Edmonton. So this was not a new discovery, but it was exciting nonetheless, because as I said before, generally carnivores are rare parts of fossil assemblages. So we're always excited when we can find one. So we felt pretty comfortable saying that this was an American lion, but because there is little information on how to reliably differentiate the toe bones of big pantherine cats, we did hedge our bets a little bit, and we said that it is what we call Panthera CF, Panthera atrox, which basically just means it's a pantherine cat. It looks like Panthera atrox, but we don't want to be overconfident because we recognize that our understanding of toe bones is maybe a little bit lacking. We then had some specimens that were a little bit more of a mystery. So this is an ulna, so that's a bone from the forearm. It's the one that kind of makes your elbow work like a hinge. And it was originally identified as the saber-toothed cat Smilodon fatalis. So of course, this excites me as the saber-toothed cat studying person. But once we started comparing it to the bones of other big cats, we found it looked a lot more similar to a pantherine cat, like a lion or a jaguar. It didn't really look like all of the Smilodon bones that I saw. Instead, it looked like these sort of more recognizable big cats to us today. But what was confusing here is that it's smaller than any of the ulnae known from the American lion, but it's bigger than any jaguar that we've seen evidence for. So it kind of fit in this weird little middle zone where, to our knowledge, no cats in the southern parts of North America occupied. So what we did is we looked to the other North American pantherine cat, which is the cave lion. Now, cave lions are found across the continent of Eurasia, and they are also known from the sort of northwestern part of North America known as Beringia. This includes the state of Alaska and the Yukon Territory. But to our knowledge, the cave lion was only isolated to the Beringian areas and had not ever been documented further south. So lo and behold, once we compared this specimen to cave lions from the Yukon, we did actually find that they were very similar in size. What was a little bit unfortunate is that the most informative parts of the bone were broken off, so we had to make this a very tentative assignment. This would be another case, just like the Smilodon that we were interested in the first place, of a very significant change in our understanding of where these species lived. So we wanted to be very careful with this, this taxonomic identification. So this is a tentative assignment. Once again, we have that Panthera CF, Panthera spelaea. It's possibly a cave lion. And what I'm really hoping is that we'll be able to find more specimens, whether in the Medicine Hat area or elsewhere in Alberta, that will help us test this hypothesis that the cave lion may have actually moved further south than Beringia at times. And there is some sort of tangential evidence that supports the fact that these cave lions may have gone that far south. So in 2013, Julie Meechan and colleagues also suggested that the Beringian wolf ecomorph was present in the natural trap cave in Wyoming, so south of where we're looking at in Alberta. And these researchers proposed that the wolves got there by migrating south between the two major ice sheets that were existing in North America at the time. And if you look at the route that was open during this time period, you see that it passes right through the Medicine Hat area. So it does seem that other northern taxa may have migrated south below the glaciers. It does seem that the route they most likely would have taken to get there would have led them through Alberta. So it's not completely impossible or not completely unlikely that these cave lions may have done a very similar thing. And then lastly, for the cats at least, we had a very exciting find. So this was the 5th metacarpal, so that was a hand bone, the one associated with your pinky finger, of something that was originally identified as a saber-toothed cat. And when we compared it with Smilodon fatalis bones from Peru, as well as a bunch of other big cats that were found in the late Pleistocene, we found that it was a dead ringer for these Smilodon fatalis bones. So if you're looking at the photograph on the right here, A, B, C, D, and E, those are all the bone that we found from Medicine Hat. Oop. And my cat is changing our slides. So A, B, C, D, and E are all the bones that are the bone that we found from Medicine Hat that was identified as Smilodon. F and G are Smilodon bones from another population from Peru. H is a different type of saber-toothed cat called Homotherium, and J and K are from the American lion. And you can see that the bone that we found in Medicine Hat is so similar to the other Smilodon bones. It's incredible. So we felt pretty confident in assigning this to Smilodon fatalis and confirming that, yes, we did have Smilodon in Canada. Taking a quick break to throw my cat off of the desk. There we go. He knows that we're talking about cats right now, so he wants to get involved. So what this identification of Smilodon from Medicine Hat does is it expands the known range of Smilodon fossils north by about 1,000 kilometers. So it goes from what you see here on this slide to this. And it just so happens that this sort of weird point that we see here is flanked on either side by the two major ice sheets that were covering the northern parts of North America at this point. So this sort of distribution actually makes a lot of sense because we had these ice sheets that were blocking the specimen from living and being fossilized in other areas of North America. So this sort of brings us to the question of, okay, if we had Smilodon here, do we also have some of the other really well-known carnivores from the late Pleistocene? So one of these well-known carnivores is the direwolf, which is the only species that is more common than Smilodon at the Rancho La Brea tar pits in Los Angeles. So this is a new paper that we had come out just a couple of weeks ago. And it sort of takes the same format as the descriptions of the cats. So we have a left dentary, so that's a lower jaw, from a specimen that was originally identified as the direwolf, so Canis dirus or Anasion dirus. When we compare it to direwolves as well as gray wolves, we do find that it does look quite a lot like a direwolf, and it is about the right size. But unfortunately, because this comes from an old individual, and as animals age, their teeth especially wear down, we actually find that all of the diagnostic characters of this specimen are gone. So that led us to need to take a different approach to trying to identify this specimen. So what I did is I worked with a colleague, Talia Lowy Mary, to characterize the shape of our direwolf, or our potential direwolf, I should say, as well as a whole bunch of direwolves from Rancho La Brea, a whole bunch of fossil gray wolves from Beringia, so from the northwest of North America, and a bunch of recent or extant gray wolves. And what this was able to do is it was able to describe essentially the shape of all of these different bones. So it gives us the typical shape of a gray wolf and the typical shape of a direwolf, and then it allows us to statistically classify our unknown specimen and get a number telling us which species it is most like. And it turns out that our specimen is statistically highly likely to be a direwolf. So we felt pretty comfortable saying that this specimen was indeed a direwolf, and therefore we had these sort of two hallmark Rancho La Brean carnivores present in the Medicine Hat area at approximately the same time. So this means that in general, there were many more large carnivores in Alberta in the Pleistocene than there are today. And the types of large carnivores that were present were similar to those found elsewhere in North America. So that being said, carnivores are really only one piece of the story in any ecosystem. And I've been focusing quite a lot on the carnivore species of Medicine Hat, but there are many herbivores as well. So what's really interesting sort of very broadly about all of the species in the Medicine Hat area is that we have a mixture of species that are still found today, along with those that went extinct at the end of the Pleistocene. So once again, we have this list of species, and all of these species that are still alive are listed in black, and all of the species that are now extinct are listed in green. So what seems to be the case in the Medicine Hat area is that we have these really great deposits that cover the end of the Pleistocene and into the Holocene. Because of this, we can start to look at how North America went from a continent with many large herbivores alongside the large predators that hunted them to what we have today, which is an ecosystem or a series of ecosystems where we have far fewer big species, and we have ecosystems that are arguably depauperate compared to what they could be or should be. So to start looking at how things have changed over time, the first sort of thing that is helpful is to have an idea of how common each group is overall. So when we look at the abundance of taxa across all Medicine Hat localities, we see that horses make up nearly half of the specimens in our collection. And other abundant taxa include the camel, camelops, the ground squirrel Spermophilus, the mammoth Mammuthus, and bison. When we break up this analysis into the 6 different localities that had more than 50 specimens, this general trend mostly holds up. So in general, we see that horses are the most common group of animals across all localities. So horses make up anywhere between 43 to 69% of specimens at all 6 of these localities. At 4 of the 6 localities, camel is the second most abundant taxon and varies in abundance between 9 and 30%. But there are 2 localities where the sort of second ranked taxon is a little bit different. So at Mitchell Bluff, the ground squirrel Spermophilus is the second most abundant genus. This is really interesting because for most localities, specimens from small-bodied taxa are either rare or not present at all in our collections. There's a number of reasons why this might be. It might be that small taxa were not being preserved in that area at that time. It may be that they were not being collected by the researchers that were collecting the sites. It may be that they're just harder to see, and therefore, whether it's a conscious bias or not, they're harder to get, or it could just be that they were actually not present in that area. And likely a combination of some of these factors is going to be at play in most sites. What's notable too is that Mitchell Bluff seems to have been the most heavily sampled localities. So we have the most specimens from Mitchell Bluff, and that suggests that maybe with more intensive sampling at a site, you might see more and more of these small-bodied mammals. And then the last of the 6 sites, Lindo Bluff, had bison as the second most abundant taxon, with camel in third. And I'll return to why I think this is the case later. Now, unfortunately, almost all of the localities in the Medicine Hat area don't just span one point in time. They actually span multiple interglacial periods. And we don't have precise stratigraphic data for all of the specimens from each locality. And what this means is that we can't use our locality data alone to assess faunal turnover. Thankfully, 175 of our specimens do have associated temporal and stratigraphic data, so we can use these to get a really coarse-grained look at how faunal composition changed in the area through the Pleistocene and into the Holocene. So while we have a much smaller sample size than the total of over 1,000 specimens that we had here, we can start to see some interesting trends. So the first thing that we see is that horses stay relatively abundant throughout the Pleistocene and into the Holocene. Where it starts to get really interesting for me, though, is that while the camel is quite abundant through the older time periods, so through the Kansan, Yarmouthian, and Sangamonian interglacials, there's a sharp decrease in camel abundance in the Wisconsin interglacial. And this is followed by a complete loss as camelops went extinct during the Pleistocene to Holocene transition. The sort of complement of the loss of camelops is that while bison are relatively uncommon through most of the assemblage in which they're present, so they're relatively rare in the Yarmouthian, Sangamonian, and Wisconsin, and not present at all in the Kansan, they absolutely explode in abundance in the Holocene. And this brings me back to Lindo Bluff, which is the site in which bison was the second most abundant taxon. So the specimens recovered from Lindo Bluff are predominantly postglacial, suggesting that the abundance of bison may be due to the increase in abundance of this group of animals following the last glaciation. So all of this is very preliminary, but we can start to see some broad patterns in the sort of how faunas were changing throughout the late Pleistocene in the Medicine Hat area. So the first thing we can say is that Equus is undoubtedly the winner of the Medicine Hat fauna game. Horses stay common throughout the entire period and are only extirpated from North America after the Holocene starts and after we have records from the Medicine Hat area. We see that camels are relatively abundant at most localities, but they seem to be decreasing in abundance through the late Pleistocene until they finally disappear in the post-glacial sediments. So camels are sort of on a downward trajectory throughout this time period. Bison are the sort of opposite of that, where they're relatively uncommon throughout the Pleistocene, but become very abundant in post-glacial deposits. And then lastly, we have an outstanding question about what's going on with the small mammals. It seems that they may be undersampled, and therefore we can't really say at this time what was happening to their abundances throughout time. So as with any study, this has led me to think of some things that can be done to improve our understanding of the region. The first thing is that, as you saw, most of the database specimens we have do not have precise stratigraphic information. And this is partly because at this moment, we still don't have access to the field notes taken when the specimens were collected. So we're in the process of getting those, and hopefully they might have some more information on where individual specimens came from, and therefore we can start to add more points to our sort of stratigraphically controlled data set. I'd also like to identify more taxa to the species level, because one thing that we would expect to see ecologically is turnover happening between closely related species. So usually when you see sort of changes in species abundance, often anything that is ecologically similar is what's going to replace something else. And the most ecologically similar species do tend to be very closely related and usually part of the same genus. So these genus level investigations that we've done so far may not be telling the whole picture. And then lastly, I would like to go out and do some more field work at these localities, as well as new localities that might have sort of unveiled themselves in the last 50 or 60 years. And in particular, I want to focus on getting really tight stratigraphic control and improving our sample of small vertebrates by specifically collecting microfossils at any sites where that might be possible. I did get to dip my toes a little bit into doing some more field work a few years ago before the pandemic, when I went out with a really excellent crew and we started to get our bearings, we started to identify sites that will be good to target in future years, and we started to re-identify some of these old sites that were previously collected. You can actually watch some of these adventures in the second season of Dino Trails, which came out a few years ago. And with that, I would like to thank you for listening and invite any questions that people may have.
All right. Thanks, Ashley, for an absolutely fascinating talk. It's so kind of wild to think that we had all of these big carnivores in North America at this time. And it kind of gives you a different sense of a world that was like sort of similar today, but also a little bit different. So a couple of questions. So the first one is, why did things like Smilodon go extinct? Why don't we see these big, particularly these cats here anymore?
That's a really good question, and there's sort of two prevailing hypotheses as to why we don't see these big carnivores and big herbivores today. So the first is that the end of the Pleistocene and the transition to the Holocene was a time of very rapid climate change, not as rapid as what we're seeing today, but the climate was very volatile. So there are hypotheses that climate was a major factor in this extinction. The other hypothesis is that humans were migrating throughout the world, and as they were encountering new regions like North America, they were potentially hunting species to extinction and kind of taking the carnivores that relied on that prey down with them. In reality, the more data that we get seems to indicate that we can't pinpoint one exact cause. It seems to be very species dependent. Some species were more vulnerable to climate change, where others were more vulnerable to human impacts. And in some cases, it's sort of a 1-2 punch. So we're still trying to untangle the exact reasons why they went extinct. But those are the prevailing hypotheses.
So yeah, just a reminder for anyone who is watching on YouTube or Facebook, if you have a question, just feel free to pop it into the chat and we can convey it to Ashley. Another question about the La Brea tar pits. Why are there so many carnivores there where they're rare everywhere else?
Yeah, it's a really interesting thing. So the La Brea tar pits, as well as certain caves and deposits like that, are what we call carnivore traps, just because like you said, and like I was talking about earlier, they have a really ridiculous number of carnivores compared to what we'd expect. And basically a carnivore trap is anything that seems to attract carnivores at disproportionately high numbers. In the case of the Rancho La Brea tar pits, what's happening is you get these pools of sticky asphalt. And when a large herbivore like a ground sloth or a mastodon wanders into these pools, they run the risk of getting stuck. It's sort of like quicksand. It's very sticky. They have this really cool display at the La Brea Tar Pits Museum, where you like get to pull on a bar to see like what it would sort of feel like to try to get your leg out of these tar pits, and it's quite difficult. So what would happen is these big animals would get really, really, really stuck, and then they would start to call out in distress because they're stuck and they want to get out. And these distress calls would attract all of these carnivores that were hungry and looking for an easy meal. Those carnivores would likewise get trapped in the tar. And therefore, you would get all of these carnivores coming, and you might get hundreds of carnivores, maybe not hundreds, but tens of carnivores for just one big mammoth or mastodon. And when all of that adds up, you just end up with a ton of carnivores compared to all the herbivores.
Cool. Kind of tragic for the animals got stuck, but great for paleontologists.
Yes. Very good for paleontologists.
So a question, amazing talk. What were the major factors in niche partitioning between large Pleistocene cats? It is thought that the large cats were largely in competition with each other. Is it thought that they're in competition with each other?
Yeah, there does seem to be some evidence for pretty strong competition between these large carnivores. Our ability to identify what these animals were eating is good, but a little bit limited because we rely on stable isotopes, which is basically what do the herbivores eat. So any herbivore that eats the same thing is going to be sort of lumped in together. So we don't necessarily know the fine scale niche partitioning that was going on. But there is evidence that there was more breakage of teeth that was happening, which indicates that animals might have had to utilize carcasses more than they would in somewhere like the African savanna today. So that may have indicated higher levels of competition. In terms of how they were avoiding each other, I think once again, the African savanna is probably a good example of an ecosystem that is very rich in both carnivores and herbivores, where you do get a lot of these sort of direct butting of heads between carnivores. I would expect to see a lot more of that. You also may expect to see different carnivores hunting at different times of day to sort of avoid the big bads, things like that.
And sort of follow-up question to that, what was the main food source of Smilodon, if it had one?
Yeah, it seems to have eaten a number of different things depending on exactly the time period and where it was, but it was definitely eating these large herbivores. So horse did seem to make up a pretty decent amount of his diet, as well as ground sloths. Something like mammoths and mastodons were probably a little bit too big for it to be eating full-grown adults. There are evidence of another species of saber-toothed cat, Homotherium, eating a lot of young mastodons. So it was probably a mixture of things, pretty much anything it could get its paws on and its claws into. But it seems to have definitely been favoring these large animals rather than hunting sort of smaller things.
Would the decline of the camels contribute to the decline of the Smilodon, or was that not temporally correlated?
You know, it may have. The camels themselves are not as common at Rancho La Brea, and that's where a lot of these studies have come out from. So it would be interesting to look at a different population where you do see more camels and see how much their diet seem to have been made up of them. But yeah, I'm not sure. It would be an interesting thing to find out.
Oh yes, one more question about the sloths. The sloths that Medicine Hat, they're a little bit of an unusual thing to find in the prairies.
Yes.
Could you just talk a little bit about the sloths?
Yeah, so there's not much that we know about the sloths. I am certainly not a sloth expert. One of the sloth specimens has been investigated by an expert and has been sort of since sort of disqualified as a sloth, I should say. It's probably not a sloth. It may have come from a mammoth. The other one, though, does seem to be a sloth, and not being super familiar with them and their biogeography, I don't actually know off the top of my head, where else we find sloths, like sort of what the next most northern area is. But certainly I wouldn't have expected them to be there in large numbers. We certainly don't get a lot of sloth fossils. There's only a couple of them. So they don't seem to have been common in the region, although probably were present.
Yeah, that's cool. Just see here. I don't see any more questions here. So I just want to thank you again for your time and for sharing your fascinating research with us.
Yeah, thank you for having me.
Yeah. This video will be available on our YouTube channel. We'll also post the link on our Facebook page as well. And I will include Ashley's website in the post. Definitely check it out. So lots of cool information about kind of work she's done and work she hopes to do. And we definitely hope to hear more about Ice Age mammals in the future. Maybe something from Northern Alberta. If you
Oh yeah, that'd be
happen to
great.
get the time. Yeah. Right. Well, thanks again, Ashley. We really appreciate your talk, and good luck in your future field seasons.
Thank you.